chapter 5, which is dealing with, as its name, as its title, extracellular messengers. And it's really going to, just like the first unit, it's going to serve as a foundation for the rest of the semester. If you look at the layout of the class, it goes chapter 5, extracellular messengers, and then chapter 6, hormones, which are a form of extracellular messengers. But if you look at the way I've moved the course, we're going to move the endocrine system to the start of the third unit. So where I'm going is clearly the chapter 5 material is relevant not only to unit 2, but unit 3 and unit 4. And I'm just flipping it so that you can get all of the nervous system into a single exam, if you will. Okay, so it's the nervous system, which does rely upon extracellular messengers. But it's difficult to go into the nervous system without having an idea of how cells kind of talk to one another, if you will. Does anybody know where this is? Nobody? It is the Alamo. Good job. Whatever. In there? Yeah. Just remember it. All right, I'm going to spend some time on this image. It's just going to give us a lot of information, so there should be a little bit of space around the images if you have a handout, and you can just kind of take notes on that image or another piece of paper, whatever works for you. But for me, images tell a lot, and even though these images are very simple, they still give us a lot of foundational information. And what these images are doing are trying to summarize the different types of extracellular chemical signals. I could say messengers, ligands, throw in the word, if you will. And it just kind of summarizes them or simplifies them. But we should see a common theme, and we'll point it out here, and then in our notes just formally address what's going on. And the point is, the messengers are where? On the outside of the cell, which is receiving the signal. And so what we're going to, you have an idea now. The cell that's going to receive the messenger probably needs to have some form of what? Receptor. Okay? And that's what we're going to unfold in a little bit better detail. Where might that receptor be, and what might it do? That's what we're going to cover here in chapter 5. And a little bit more technicality than just saying there's a receptor and it receives a messenger. So if we look at the different types of extracellular messengers, the first messenger that we're going to utilize this semester is called a neurotransmitter. So I'm going to go down to the second row, first image on the left over here. A neurotransmitter. And if you look at that lower image, we can see two cells, if you will, talking to one another. It's not verbally, but how? It's what? Chemically. Basically a chemical talk, if you will, from one cell to another. And so in other words, one cell will make the message, the other cell will do what with it? Receive it and then carry out the message. Very good. In this case, the messenger we call what? We call it a neurotransmitter. That's just a very more technical term. Instead of just saying extracellular messenger, well, I know, yes, it's extracellular, but it came from which type of cell? A neuron or a nerve cell. Neurotransmitters are chemicals that tell other cells what to do, but they were manufactured in a cell that's called a neuron. Okay? And that's what we'll study for the rest of this unit. So neurons release neurotransmitter. In this case, we can see the receiving cell is another type of what? Neuron. It's not always nerve to nerve. We'll learn in the second unit. You may understand how your nerves control your muscles and other factors as well. But here the thing is, can you see the blue dots, the neurotransmitter, and how it's outside of the nerve cell which it's controlling? The term that we're going to use to describe the cell which receives the message, you can be real generic, you just call it the target. So label this cell down here the target. The target is the cell that what? Receives the extracellular messenger. And it has specific what? Very good, it has specific receptors. So in this second row, first image, we can see the target cell is receiving an extracellular messenger called a neurotransmitter. The type of neurotransmitter we're not going to be specific yet, but there are examples of like acetylcholine, dopamine, serotonin. Have you heard of those? Epinephrine. So we'll get more specific. Right now we're just saying a chemical that comes from a nerve is called a neurotransmitter. One major type of extracellular messenger, but not the only. Another important extracellular messenger we'll study this semester are called what? And you're probably familiar with this word. Hormones. Okay, so looking at the first row, first image, we can see hormones can also serve as extracellular messengers. Unit 2, we're going to go bananas, if you will, on the endocrine system and different types of hormones like testosterone and estrogens and adrenocorticotropin and all kinds of good hormones. Right? But right now we'll just say they're hormones. They're extracellular messengers. What type of cell would make a hormone? Endocrine cell. So over here on the top left of the image we can see an endocrine cell. It produces hormones. And now we can see the big differences between an endocrine cell and a nerve cell. What's different in this top left image? Very good. The hormones not only go into the interstitial fluid, like we saw the neurotransmitter, but it also goes into the blood. So hormones are circulated in your blood. So they're far away from the target cell, most likely. So if you think about, say, the pituitary gland, far away from the ovaries. The hormones from the pituitary have to go into the blood to reach their target, wherever they may be, which we'll learn about 2 weeks. Okay? So let's go ahead and make, if you didn't write it down, let's just make a point to illustrate that. Hormones go into the blood. Neurotransmitters, do they go into the blood? They have a short distance, a little bit of interstitial fluid which they have to diffuse across. But they're adjacent, or right next to the target. Endocrine cells that release hormones are probably really far away from the target. And so therefore the only mode of connection, if you will, is the circulatory system. But either way, the target cell is going to have a what? A receptor. Why don't you just draw a square receptor on your nerve cell, target cell, and draw another one in your endocrine target as well, acknowledging that there has to be a receptor. Include neurotransmitters from cells called neurons. The other are called hormones from cells that are collectively called endocrines. And then if we look in the first row, the second image, it messes us up. Okay, if you go on our technicalities, we can see what type of cell. Nerve cell releasing its transmitter into the bloodstream. There are some exceptions. It's kind of like a conglomerate, if you will, where some nerve cells have the ability to release their neurotransmitters in the blood. This image calls it hormones, but I want to be technical. This is a technical class, so I want you to add the prefix neurohormones. N-E-U-R, neurohormones. Some nerve cells have the ability to release their neurotransmitter into the blood. They're still going to reach a target cell, which still needs a receptor. So you don't get confused later, let's list some examples. Have you heard of the adrenal gland? The adrenal medulla releases epinephrine and norepinephrine. You've heard of those. Maybe you called it adrenaline and noradrenaline. technically those are neurotransmitters, but they go throughout the whole body in the blood. You heard of oxytocin? Comes from the posterior pituitary, which is nervous tissue, but it goes into the blood. Targets, say, the uterus and other structures as well. So there are some exceptions where nerve cells can release their neurotransmitters into the blood, but because they're in the blood, we'll call them what? Neurohormones. But the target cell is still a target cell and it still expresses specific receptors. Those are the three major extracellular messengers that we are going to study this semester. Neurotransmitters, Unit 2, hormones and neurohormones, Unit 3, and then 4 as well. They aren't the only types of extracellular messengers, though. There are other types of messengers which act as, it's not written on here, so I'll tell you in just a moment, but you can see illustrated in these images where one neighboring cell can release a messenger and affect its neighbors. And so it's kind of like neurotransmitter, except for these chemicals aren't what? They aren't neurotransmitter. Okay, they aren't nerve cells releasing them. This type of signaling, where you can see the outward arrow, where one cell is affecting a neighboring cell, is called paracrine. P-A-R-A-C-R-I-N-E. Paracrine. The paracrine messenger is not going into the blood, just hanging out in the interstitial fluid, and its neighboring cells happen to have receptors. And those cells are communicating together in paracrine signals. We see another type of signaling, in this image. Anybody see it? What's another type of extracellular messaging? What is it? Did I hear it in the back? That was hopefully. Can you see a different looking arrow in that last image? What's happening? What's it doing? It's targeting itself. Some cells have the ability to release an extracellular messenger and still express the receptors. In that case, it's actually targeting itself. Term for self is, what's the prefix for self? Auto. And so we just call it autocrine. Where a cell releases an extracellular messenger and expresses the receptors and therefore responds to that extracellular messenger. So it activates itself. You'll see more of these examples in the nutrition, like regulating the digestion, and then also in the immune system, where you'll see some of these more characteristic than where we're going this semester. What questions do you have over these major types of extracellular messengers? Neurotransmitters, hormones, neurohormones, and also paracrine or autocrine signaling. All right, good. I want to spend some time on this as well, and actually I think it'll be it's easier for me, hopefully it's easier for you, for me to discuss with you guys where we're going to go, and then we'll go back and fill in the notes. So again, you might want to use another piece of paper or just give me your attention or however you want to work with it, but I wanted to discuss this. Looks simple here, but you can add a lot of detail here. Okay? We're going to have to build upon what you learned in Unit 1 about where different molecules are manufactured in a cell. We were sure to differentiate where proteins that are to be excreted, like, say, an extracellular messenger. If I want to produce a protein that's to be secreted, where is it manufactured? The rough ER. Made in the rough ER, processed in the, using ribosomes, processed in the Golgi, wrapped in vesicles, and at some point it is, what is the term? Exocytose. It is if we were looking at the vesicle releasing its contents, it's exocytose. And so we're putting a connection here. I'm talking about an extracellular messenger. If it's protein-based, we're going backwards. It was originally made in that cell that made its, what? We said the protein extracellular messenger, whether it be a neurotransmitter or a neurohormone or a hormone, it was made in that nerve cells, hormone cells, whatever. You said what? Rough ER. Our proteins are only messengers? No. Other molecules can be messengers, and the big other major group of messengers are lipid-based. Where are our lipids manufactured? In the smoothie arm. And so depending upon the type of messaging molecule, we can already tell where it was manufactured. Lipid-based molecules are lipid-based, so can we keep those trapped in a vesicle? No. So now we're beginning to got to take into consideration if something's lipid-soluble, how does it get out of the cell, versus if something's protein, right, or water-soluble, how does it get out of the cell? So we've got to add all these features about it. And then if we know how something is, where it's made, an idea about what it's soluble in, we also therefore know if it's more soluble in water, which we have everywhere, or a lipid, which is in our what? Different types of our what? There's a lot of lipids where? In the plasma membrane or any other organelle, right? Membranes, valves, structures. Okay. So we've got to build from unit 1 is where I'm going. And we can categorize our extracellular messengers, hormones, neurotransmitters, neurohormones, whatever, based on their solubility. Whether they are more soluble in water, like what type of molecules might be more soluble in water? The two options we just gave. Actually, lipids are water insoluble. You've ever tried to mix water and oil? They separate. So what is more soluble in proteins? Are the two options which we studied. Proteins are more water soluble. And so those types of molecules we can consider to be hydrophilic, water soluble molecules. But not everything's equivalently water soluble. Some things are more soluble in, we said, fats or lipids, like what part of the cell? Like the membrane. And particles that are not very soluble in water, but more soluble in lipids or fats, we can just call them lipophilic. Okay? Philic tells us what? Loving. Lipo means fat or lipid. So in other words, our molecules that we're going to study this semester are either more water soluble or more fat soluble or lipid soluble. If something's more water soluble, it's probably, what is my abbreviation here? Amino acid based, or what word have I used so far? Proteins. Our proteins, whether they're tiny little amino acids, okay, or smaller peptides, like decapeptides and whatever, or large proteins, are most likely going to be water soluble. Where is their water? So in other words, where would they dissolve? In the cytoplasm, interstitial fluid, and even our third, what part? She said extracellular fluid, we listed already the interstitial fluid. The other component of our extracellular fluid is the blood plasma. We've got to kind of think globally now if we're moving a particle from a cell across a little synapse, or a hormone from the brain to the ovary or something like that. We've got to think about how's it going to get there. So let me recap. If something is more water soluble, it's probably amino acid based, however big it is, 2, 3, 4 amino acids or hundreds of amino acids, like I think insulin protein, right? It's going to be considered to be water soluble. And what we said, there's water inside the cell and there's water outside of the cell. However, for that particle to go from inside to outside of the cell, we've got to get across the cell what? Membrane, which is full of fat, lipids. Okay, it's lipophilic. Lipids. We already know if I have a protein inside of a cell, the only way to get it out of the cell is to how? Well, you could do a channel. We haven't studied any channels for proteins though, so we have a vesicle. Okay, it is a carrier, but it's a vesicle that releases the protein outside. Once a protein is released outside of the cell or exocytosed out of the cell, it's in the interstitial fluid and it's what in the fluid? It's soluble. If you have a water-soluble protein out in the interstitial fluid, it's trying to reach its what type of cell? Target cell. Target cell needs to have what? Receptors, which we know are also proteins. The question is, then where would the receptor be located for a water-soluble messenger? We've really only seen one option so far. Where would the receptor be at a target cell if the messenger is hydrophilic? In that target cell's plasma membrane. In that target cell's plasma membrane. Because the protein cannot do what at the target cell? It's there. Is what type of solubility? Water soluble, not lipid soluble, so it's probably really big and it can't what? It can't, right, I'm going to modify your word. You said it can't be released, but it can't, what is the word that we used last week? It can't diffuse across the cell membrane because it's big and charged, okay, water soluble. So what we're going to learn is a hydrophilic messenger, we know where it was made, we know how it's released, but at the target cell receptors have to be in that cell's membrane because there's no way to otherwise let that particle diffuse into the target cell. It's too big, it's too charged. You didn't realize you learned all that last week, did you? You did learn all that last week. We're just putting it in application this week. So are all messengers protein-based? No. Some messengers are lipophilic or lipid-based. Jason, you probably are more familiar with the word, I think you use the word steroid. Our steroids are lipid-soluble or lipophilic. Where did you guys say lipid-soluble particles originate? In a cell that makes them, they originate where? In this smoothie arm. There's a lot of different types of processing that can happen. But once the lipid-soluble messengers are made, how do you think they get out of the cell which made them? How do you think they get out? Do you think we need to exocytose them? No. They just simply, actually most of them simply diffuse out because they are lipid soluble. So our steroids, basically, they're lipid soluble. Once they're made, they diffuse out of the cell which made them. But they're now in the extracellular fluid that has a whole heck of a lot of what in it? Water. So if you think about hormones that go into your blood, and these things are lipids, logic doesn't make sense. I mean, if we try to reason it through. What would these things do if we tried to send them into the blood as is? Say what? They would repel the water. And have you ever seen the oils and the vinegars like at a dinner table or something? Have you ever seen how they form little, like if you shake up something and you see little spheres of the globs, right? Do you think spheres and globs of fat are good in the blood? estrogen blobs in our blood. What could we do to something that's lipophilic to make it able to transport and say our blood really well? What do we do? Well, the good option, you say metabolize it? Catabolize it, so break it down, which is a good option, but if we break it down, then it's no longer the messenger. So at some point, you're right, it will have to be catabolized. But before even then, what has to happen to it? If it's lipid soluble, I want it to be what? Water soluble. What could I do with it? I think you were on it a minute ago. We need to give it a what? A carrier that happens to be water soluble. So I could bind my lipids with things that are water soluble, like proteins. Our lipid soluble messengers hitch a ride with water soluble proteins. And that's all they do, is they just carry the lipids. That's all the carrier does. The whole point is to get the messenger to a, what kind of cell? A target cell. The target cell needs to have a receptor. Probably the receptor is located where? We think plasma membrane, because that's all that we've known. But the carrier lets go, the thing is still what type of solubility? Lipid soluble, so what could it do at a target cell? Just diffuse in. Where then it would interact with its what? Intracellular receptor. Your lipid soluble receptors are inside of the target cell because the molecule is actually lipid soluble. It can just simply diffuse into the target. And so what may be new to you is that not all receptors are actually in the membrane. Some are actually inside of a cell's what? Cytosol. Most notably if we're looking at our lipid soluble messengers like estrogen, testosterone, whatever, the receptors are inside of the target cell. Cytosol. So, y'all are looking a little bit like I'm making stuff up. But this is, I think, to me, a little bit easier way to understand something. If something is water soluble, we just gained a ton of information. We know it's probably what type of molecule, probably a protein. And if it's on the outside of the cell, well, then we can presume it was made where? In the rough ER. We also know then how it was released. Exocytose. We know that if it's exocytose in the extracellular fluid, it's soluble, so it doesn't need a carrier. It's good. We also know that the target cell needs to have a receptor, and that receptor should be located where? In the membrane of the target cell. Because the protein is not, it is water soluble, it's not lipid soluble. It just can't diffuse in. Conversely, if we look at something that's more lipid soluble, we also know that it's manufactured where? Probably in the origins of the smooth ER. Once it's made, cell has difficulty holding onto it, so it just first it diffuses out of the cell. Most of them diffuse out first and then attaches to a carrier, and that's all it does. Gives it a ride. And then it has to diffuse into the target, where then it would expose to its receptors. Just by saying hydrophilic or lipophilic implies 30 minutes of discussion. Okay? And so that's why I think it's easier to look at something. Is it more water soluble or is it more lipid soluble? We know that the world's not black and white. It's not always just water soluble or just lipid soluble. but it's easier if you just can understand things in that category. And then once you understand either or, then you can vary the gradient, if you will. Okay? And so what we'll do is we'll go back and review this information. Right now we said if something is more hydrophilic, it's more water soluble, probably a protein. If it's water soluble, you can actually call it lipophobic because it's repelled by fat or lipids. And conversely, in our body, if something's not more water soluble, it's probably more lipophilic or lipid soluble. So all of our steroid hormones and another one called thyroxine, which we'll clarify in, I think today actually, why is an exception. If we look at, this is going to be a repetition for which I just mentioned. Okay, so this will be a review. We said if you understand how something is soluble, if it's more water or lipid soluble, we know probably where it was manufactured or synthesized. We also know if it needs help being transported in the extracellular fluid, and most specifically the blood plasma, is that's a concern. And I have 3 that exerts its effects at the target cell, okay, because we know where the what is located at the target cell, the receptor. As well, we know even though if we're looking at multiple molecules and they're all lipid soluble, they're all going to have slightly different structures, but have profound, functional differences. Classic example is testosterone and estradiol, which is a variant of estrogen. Testosterone, we think of what? Male, and estrogen, we think of female. And if you've ever looked at their structure, you notice they look extremely similar to one another. You may remember this 4-ring structure of cholesterol in the membrane. It serves as a template for our steroid hormone. And it can be processed. Okay? We see this hormone called progesterone we'll come back to in Unit 3. It can be processed to androgynous type of precursor, androstenedione, which is a precursor for testosterone. Notice the 4-ring structure is retained. We're just processing the cells and some other projections. And look at testosterone. It's the precursor for estradiol. If you look at the structure, they're very similar, but if you go through and nitpick it, there are some structural differences, which gives us obvious functional differences. So structure dictates function. Everybody get that right on their test? If you didn't, I'm going to auto-fail you if you're out of here. Eject button. All right. So this is reviewing what I was mentioning and we were discussing. We learned that if something's more hydrophilic, we know that it's going to be made similar to any other protein in the cell like we studied in Unit 1. In addition, they're not just randomly made. We know, if you remember from your Gen Bio classes, the protein needs to be made, it's transcribed, and then there's going to be something initiating it being made. So that's what that second statement means. It has to be initiated for it to be synthesized. We know that since it's an extracellular messenger, it's synthesized by the rough ER, and this arrow is implying a good day's lecture, right? He says processed by manufactured rough ER, processed, right? So a lot of people just memorize this, but I'm just trying to recall your memory. They're applying now Unit 1 material. It's made at the rough ER, processed, modified in the Golgi, where it is packaged in the individual vesicles and stored until it's released by the process, which we learned in Chapter 4, is called exocytosis. Once our lipophilic molecules are exocytosed in the extracellular fluid, they're water soluble, they're dissolved is the term. Because extracellular fluid has a high proportion of water. So whether that be the interstitial fluid or the blood plasma, these messengers are water soluble. We also noted that at the target cell has a receptor, and the receptor is in that target cell's membrane. what we'll finish up in this chapter is what do those receptors do. So we'll come back to that. But and I'll mention them really briefly right now. Those receptors do primarily one of two things when a hydrophilic molecule is present. So, for example, where we're headed in Unit 2 material, let's think about our neurotransmitters, like acetylcholine, you'll become really familiar with this semester. It's hydrophilic. It binds to a membrane-bound receptor, and it does one of two things. The thing that it does is its target cell opens up ion channels at the target cell. So acetylcholine binds to a target cell receptor, and that receptor opens or closes for specific ions. If you were in lab yesterday, what does that do? If the target cell opens or closes an ion channel, what's the net effect? Potential changes. Electrical voltage happens at the target cell. So we'll highlight focus is what I'm trying to say on these options, where a messenger just changes the permeability of the target cell in a potential. As well, some of our systems are much more complex in that they activate what's called a second messenger system, which is, you can imply, is going to activate a whole other round of messaging. And we might get to those today. If not, we'll cover them in better detail tomorrow. So because the receptor on a target cell, when receiving a hydrophilic messenger, the messenger will either cause a channel to open or close at the target cell, or activate a more complex response within the cell. If we were to contrast, just show an image. Figure 5.7a is illustrating, kind of summarizing what we mentioned. A target cell, the messaging cell releases its molecule, its hydrophilic ligand. What color is the hydrophilic ligand in this image? The blue structure. We can see it was originally in a vesicle, so it's being exocytosed. Here we can see it diffusing out of the cell, out of the interstitial fluid, and actually, in this case, into the blood. So this tells us this is a hormone. And it's in the blood, and we don't see anything extra affixing to it. So it's dissolved. At the target cell, if we're looking at a hormone, it has to get out of the blood and then diffuse across to the target cell. So here are two options for some target cell. On the far right, we can see the membrane really well. And on the far left, we can see another scenario where the membrane is green in the background a little bit. In the left image, what color is the receptor? What color is the receptor in the far left image? The red structure. In the far right, the receptor is colored the purple structure. The yellow thing is the messenger. This is going to, if we can have your attention a little bit better to help labs is really lasting through the semester. Notice how this receptor is bifunctional. It's a channel. What kind of channel is it? Your lab yesterday, it's gated. Not only is it a gated channel, it's also we're calling it a what here? It's a what? You say channel? Is it, it's not voltage gated. What's opening or closing? Good option, though. What's opening or closing it? What is this thing? We're talking about open or close it. This whole unit is about these things. The messenger. So in lab yesterday, we called these chemically gated ion channels. So it's the chemical that opens or closes it. Or I could say the extracellular messenger. It would cause a voltage change, but it's not the voltage that actually opens it. Does that make sense? Just nod so she'll move on. So our extracellular messengers that are hydrophilic could open or close directly an ion channel. And we'll repeat these throughout the semester when we look at them in better detail in different applications. Or, in the second scenario on the right, our extracellular messengers that are hydrophilic may bind to a receptor, but if you look at the receptor, it doesn't just open or, it's not a channel. it actually activates another pathway. It does this, then that, then this, and that, then that, and that. And then something happens on a big scale. So the takeaway at this point is an extracellular messenger that's hydrophilic binds to receptors that are where at the target? At the target cell's what? At the target cell's membrane. And it may, when present, may cause opening or closing a channel or activating a second messenger. There are many second messenger systems. The one that we will study include the cyclic AMP pathway. There are others, kinase pathways. There's lots of different messenger pathways. We won't study them in this class, but when you go to biochemistry and or certainly cell biology, you'll look at the variation of these. But we'll look at the common thing, one thing, then another, then another, then another. They'll drive you crazy if you let them. We contrast that to our lipid-soluble particles, as we reviewed earlier. We know that cholesterol is going to be the template molecule, or the precursor molecule. If I use the word template, does that conjure up, does that make sense a little bit? Template? You ever had a template for something? It serves as the starting molecule. And so once cholesterol is available, we can use it to make other steroid hormones. The way that it's manufactured is enzymes will take cholesterol and modify it. And that second bullet, it depends on which cell type you're in. So for example, the ovaries probably make which hormone? Estrogen. The testes would have enzymes that manufacture testosterone. Okay. So even though cholesterol is the same precursor, each cell type would have its own enzymes to make its own hormone. I have the word, the third point, it says immediately passing through, but really we should be more concise and say that the molecules do what once they're made? They diffuse. They diffuse to the plasma membrane, and now they're in the extracellular fluid, where we're going to have to address in just a moment. But because our lipophilic molecules are lipophilic, we can't store them. We can't keep them in vesicles and wait till we need them. So as soon as they're made, what do? They diffuse out. So they're continually needing to be manufactured. Which makes sense, ladies, if you ever had or even gentlemen too, if you take the thyroid pill, somebody, I don't want to call you out, or ladies, if you take the birth control pills, you have to take them how often? Every day, same time, because these particles are lipid soluble. They're always diffusing and out, right, too. Okay, so we can't keep them. So what brings us to the last 2 points is because we can't store the mature steroids, as soon as they're secreted, or manufactured, they're secreted. But once they're secreted out of the cell, they're in a very watery environment, which they aren't very soluble with. Okay? So they can't dissolve well in the extracellular fluid. So what we discussed is that our lipophilic molecules travel better in the blood bound to transport proteins. Without getting into specific transport proteins, some transport proteins are made just for a particular lipophilic molecule. But most of our lipid-soluble molecules just bind to proteins that are already in our blood. Have you heard of like albumin? Blood albumin? It's a big, huge protein that can bind lipophilic molecules. It just is carrying them in the blood. You're also aware, though, that protein is just a carrier. It doesn't affect the functionality of the hormone. And so that hormone has to unbind from the protein so that it can do what at the target? It can do it. Dissolve in that cell's membrane to get into its cytoplasm. Right? So our lipophilic ligands, they will diffuse into the target cell right across that plasma membrane. And as we discussed a moment ago, the receptors are inside of the cell in one of two locations, either the cytosol or the nucleus. Lipophilic molecules, because their receptors are located in the cell, are going to have a more direct effect within the target cell. And eventually, they're going to have their actual target at the nucleus. What's in the nucleus? DNA. So basically, in a nutshell, our lipophilic molecules actually promote transcription, or maybe cell division or something. They're going to have an effect directly, described right here, gene activation or protein synthesis. So they have a direct effect on promoting gene expression, is a better way to say it. So if a cell wants to make insulin receptor, it has to be initiated somehow with some steroids to promote that. An image might help a little bit better. So if you make reference to figure 5.7b, we can see here some, in this case it's okay to call it an endocrine cell. Our messenger is colored which color in this image? Yellow. We don't see it vesiculated, we don't see it bound, right? You see how it's just diffusing out of the target cell? And it's diffusing into the blood, where some of it may just dissolve, but not very much. The bulk of it has to be bound to a, I wouldn't use the word receptor. It has a similar effect, but we'd better call it a what? A carrier or a transporter. If you use the word carrier, it doesn't imply an action. If you use the word receptor, it's going to imply something's going to happen. And so the carrier or the transporter just transports. It's like the city bus. It's like if I were to go get on the city bus, the bus takes me to my work, but unfortunately it doesn't do my work for me. Okay? So I wouldn't call the bus the work, I would call the bus the transporter. Does that make sense? All that? Yeah, so the question was, how does the messenger get loose? And we really won't put it into full practice until chapter 15, 16, 17, around there. But what happens is, as your blood moves through your body, we kind of talked about in Unit 1, temperature changes, metabolism, oxygen levels are dropping, and everything. As you get into tissues, the relative oxygen drops, which makes sense because we're using it in which organelle? Which organelle is using our oxygen? For what? Well, the lungs provide the oxygen. Which organelle is using the oxygen? The mitochondria at the end of which process? The electron transport chain. Okay? So, relatively, when you get into your metabolically active tissues, oxygen's dropping, carbon dioxide is going up, temperature's going up. We'll add in there, those also alter the pH. All of these factors, we change those factors, we change the shape of our proteins, and the proteins let go. Change the function, and it lets go. So it's kind of complex. It's not magical like the bus, like here you go. But there's factors where the blood in our capillaries basically change the slight structure of the protein, which affects how they bind to the molecule. So a good question, illustrating that yes, the carrier has to let go, because more often than not, the carrier, that's all it's doing. There are some exceptions, just like everything else, but generally the carrier, that's all it does. This is summarized in the image that I have for you in your handout. Our messenger, our lipophilic messenger, is grossly enlarged, so we can kind of see it. Then here's our target cell. The target cell membrane's in the yellow, cytoplasm in the beigey color, the double membrane nuclear envelope in the nucleus. So you kind of see where we're at in this case. Our lipophilic molecule, we can't see its carrier, we presume it had one, but it's diffusing. Maybe you want to add that in there, that it's diffusing. You know, that it's diffusing across the membrane and actually through the cytoplasm and into the what? Into the nucleus. At this point, we've not seen it collide with its receptor. We can see in the nucleus, the steroid hormone can bind with its receptor. When it binds with its receptor, the receptor becomes basically activated without having to go into much detail. You see how the receptor changed shape, changed function, and now the messenger and its receptor can bind to another receptor on the DNA. So the steroid hormone binds its receptor and then binds to another receptor on our DNA. What this does is promote this process called transcription. I'm going to try to purge this. Transcription makes different types of messenger RNA, or different types of RNA, and most likely messenger RNA, which can then be translated into a protein. Whether that translation occurs at that cell's roughie or the cytoplasm, I don't know. They were being super generic. But the whole point is, our steroids promote protein synthesis, basically. But it's not real simple, right? We've got to do one thing, then another, and another. You should be able at this point to see a big difference between a hydrophilic and a lipophilic molecule, as far as where it was manufactured, how it gets out of the cell which made it, if it needs help being transported in extracellular fluid, as well as where receptors are located in the target cell. And now you have an idea what different receptors do. Hydrophilic bound receptors open or close channels or activate something called a second messenger system. Our lipophilic molecules do what? Promote what? It's in the picture for you. DNA may not necessarily be DNA synthesis. I'm trying to think of some. Sometimes I'm sure they do trigger replication, but more transcription, which makes not DNA but makes RNA molecules. So steroids promote make a new protein. Just got a couple of extra images to illustrate how our lipophilic molecules, illustrated here with the letter H, need a carrier. With a carrier, in this case, you can see it letting go of the messenger. The messenger diffuses into the cell. This time the receptor is located where? In the cytoplasm. And then the receptor and the messenger go together in the nucleus. Okay, so either interaction is feasible, either the cytoplasm or the nucleus. But the net effect is still what? Transcription and translation. But our lipophilic molecules promote protein synthesis. Before I move on, at the end of, can you turn your page? I'm going to clean my hand up. One more time. One more time. On page 6 of your guided notes, if you're using those, at the top it says on your own. And it summarizes what's taken me an hour to talk about. So tonight, or this afternoon, fill that in so you don't lose any ground that you've gained today. Okay? So on your own, it just summarizes everything that we've talked about. So go ahead and complete that on your own. I will not go through it because I just went through it for an hour. And we're going to take another hour to go back through it. Okay? So certainly if you have questions, ask me, but I'm not going to formally go through it. And so it took an hour to go through that, talking about two types of solubility, hydrophilic or lipophilic. It's really generic. We said it's more water soluble or more lipid soluble. Of course, we could get more specific, and your book does, and it talks about not necessarily the solubility, but actual structural classification of our different extracellular messengers. And our extracellular messengers in more technical detail can be called amines, polypeptides or true proteins, glycoproteins, or steroids. And what we can do is apply the solubility. So go through each of these lists of 4 and identify whether they are more hydrophilic or lipophilic. So of those 4, you're going to identify are they hydrophilic or lipophilic. So just write in the margin what do you think each of those are. what type of solubility each has. Check yourself. Your amino group molecules probably hydrophilic. Very good, because they're amino acid based. I'm going to come back to that. There's always exceptions to everything. But the high proportion of them are hydrophilic. Polypeptides and proteins, hands down, we better say hydrophilic. Glycoproteins, proteins of the sugar water soluble or fat soluble? Okay. Water soluble. Okay. You drink sweet tea? We know for sure sugars are water soluble. So our glycoproteins are hydrophilic, and our steroids lipophilic. The exception for amino acid based messengers that is not hydrophilic is thyroxine. Okay, so circle it in your group. Your handout. This is the exception. Thyroxine is an amino acid based messenger or hormone, and it is actually lipophilic. It needs a messenger and binds to intracellular, or it needs a carrier and binds to intracellular receptors. Make sure to point that out because it'll be one example we use quite a bit in the endocrine system. At this point, I'm not going to hold you accountable for the different hormones and messengers, but they're just examples. If you've heard of NE, stands for norepinephrine, epinephrine, melatonin. You've heard of those maybe? Those are amino acid-based small, most of them are hydrophilic molecules. You've heard of oxytocin and insulin. Those are larger proteins and water soluble. You've ever heard of luteinizing hormone? Follicle stimulating hormone. Those are glycoproteins and some steroids that you're familiar with, testosterone, estrogen, cortisol, or lipophilic. The things that we've already described as the target cell are collectively called signal transduction. I just didn't use the word yet, so I use the word and repeat what we learned. The whole point of an extracellular messenger is to initiate the target cell to do something. What the target cell does collectively can be generalized as signal transduction. So once an extracellular messenger activates the target cell, the events are called signal transduction. We've covered them already, but we'll come to the notes. What might a target cell do in response to a messenger? We said, what were the options that we said would cause? Maybe transcription and translation. That I let you know. What else? Activate a second messenger or open or close channels. Okay, so we're just going to repeat that information. The receptor is going to do something, and those are the primary things that happen when a target cell receives a messenger. The ligand-bound receptor. The ligand we learned is just a generic word for the messenger, whether it be hydrophilic or lipophilic. That's the first message to the target. And we said that will open or close specific ion channels, activate a second messenger, or have an effect at the genome. Last statement that's almost hiding down here is huge to us for the rest of the semester. So maybe you want to put a star by it. The response of the target cell depends on the target cell in a specific way. What am I pointing to? To its receptor. The target cell and the receptor are going to dictate the response of the target cell. What I mean is, epinephrine doesn't cause the exact same thing to all billions of cells, or trillions of cells in your body, right? Because not all trillions of the same cells have the same what? Receptor. So we're going to look at some variation in responses. In addition to just acknowledging that a target cell has to do something, we've got to take into consideration the magnitude of response. And so if I'm trying to activate a target cell, we need to take into consideration, I have the word blood levels, and I really need to just fix this. I'm going to omit the word blood. Excuse me. Probably messed that up. Just the levels of the ligand, the extracellular messenger. Okay? Whether they are in the blood or in the interstitial fluid, whatever the case may be, the proportion of that ligand is going to dictate how the target cell responds, right? If there's more of the messenger, the response to the target cell is probably what? Which thing? More, right? If there's less of the messenger, the response would be less. Okay, so it's directly related. So we've got to take into consideration how much of the messenger is available. But we've also got to take into consideration how many receptors the target cell actually has. And that's also going to be proportionate. If a cell has more receptors, the response is greater. And if there are fewer receptors, the response is less. Okay, so it's proportionate. Something that maybe you've probably not thought of before is the affinity of the receptor. What does that mean? How much and how well it binds to the receptor. So yes, maybe a ligand binds to the receptor. Does it bind well and quickly? Or is it kind of slow and delayed? That's going to affect how the target cell is going to respond to a messenger. And what we need to do is understand then, where the heck do the receptors come from? Well, our receptors are which type of molecules? Receptors are, what are receptors made of? Proteins. The proteins have to get transcribed and translated, right? Your cells can't go to the epinephrine receptor store. They need epinephrine receptor, they make it. Okay? And so, in order to make them, to add more receptors, the process is called upregulation. Upregulation. Upregulation is this collective process of transcription, translation, and expression of receptors, so that there are more receptors now expressed. And the target cell becomes what levels of sensitivity to the molecule? More or less? More sensitive. The big cell upregulates receptors, it becomes more responsive to same levels of hormone or messenger. You can see that simplified here between, it's a time phase, some cell, blue square cell. And we can see our luteinizing hormone receptors in the red. Under the influence of another hormone, we can upregulate the luteinizing hormone receptors, and can you see how we increase substantially? So we can upregulate to become more sensitive to the molecule, or we could downregulate. During downregulation, the number of receptors, decrease. So that the molecule becomes, so the cell becomes less sensitive to a particular messenger. Those words are, I think, okay, aren't they? Upregulation, adding what? Adding receptors. I can also downregulate, remove receptors. Some other terms we'll use throughout the semester are agonist and antagonist. We'll review these terms when we look at the endocrine system. or cause it to do what? Agonize. An agonist will do what? Mimic. Okay. So some molecules can bind to other molecule receptors, and it'll be an agonist, and so they kind of cause the receptor to do its job without that necessarily that molecule being present. As well as we can do what to receptors? We can promote them by agonizing them, or we can antagonize them and tell them to stop something. You're probably more familiar with antagonists. You've heard of like blockers. Maybe that's what they're probably where you're more familiar with molecular blockers, receptor blockers. Your book goes through some examples, and I'm not going to hold you accountable, but I just wanted to show you some examples of an agonist versus an antagonist. And some examples are morphine. Probably heard of that. Maybe had it, been sick or hurt or something like that. Morphine is an agonist, activates some receptors and causing that pain-reducing effect. And another one called an antagonist, which actually inhibits the pain blocking response. And so you would actually feel the pain in that sensation. So some molecules block or promote the receptor from doing its job. This is going to be repetitive. I really need to kind of rework this. So I'm just going to summarize the gated channels or the ion channels. And then tomorrow we'll get into the second messenger system. I'm going to have your attention real quick. Over the evening, you need to go through the reading pages. If you haven't already, I'm going to presume you haven't. We're coming off a test. For Chapter 5, everything which we've talked about today with the exception of the second messengers are on pages 124 through 145. So it's going to be really important that you keep up with your reading over the evening. But I want you to learn about our second messengers so that tomorrow's not painful. Okay? It'll be flying over you if you don't have a heads up on this. And I can't remember, but it would certainly be useful to get into Chapter 3, which is homework number 3 as well. Maybe read this. So we won't talk about second messengers today, but we'll look at these gated channels. If we look at hydrophilic messengers that can open or close in response to a ligand, we noted earlier that the receptor can also be bifunctional, and that is a channel. And it's described as being direct. So the molecule's present, the channel opens. There's nothing fancy or trickery about it. And the molecule's absent, the gate would be closed or however it's built. Okay? And it just causes the direct opening or closing. And we know that when the channel opens, specific ions can move through that particular channel. So Figure 512 will help illustrate for these fast gated channels. You can see the receptor site, you can see the channel, but you can see that it's gated. Hopefully you can see that it's gated. In this case, the extracellular messenger is present, the channel opens, and now it's permeable. Here's a big thing though. Does the messenger go into the target? No, this is huge. You realize that the extracellular messenger, that's all it does is bind and unbind. It is not taken into the target cell. But we're opening for specific ions like sodium or potassium or chloride or whatever it's called. We're going to change the you guys learned yesterday or will today. We change the potential. And we'll conclude there looking at these gated channels. All right, so thank you for your attention. We'll see you in lab today, or if you're in lab tomorrow, you'll have your lab quiz number 2.